Okay, so waited the traditional couple of minutes for people to solve various technical gremlins. And I'd just like to welcome you all. And yes, my name is Stuart Newman. I'm the CEO of Therapeutic Innovation Australia. And this is, as I said, the first ever TIA webinar. And I mentioned that so I can, in case there are any more technical problems, I can get my excuses in early. So way back in February 2020, which seems like a different country now, we had a meetup of all the various TIA facilities in our consortium and decided that we really need to get ourselves some more of a collective profile to make sure that the accessibility of the capabilities and the facilities are more widely known to the community. Because Australia, as we will all be aware, is terrific at research and has strong research infrastructure, and it is accessible and well maintained. But to access something, you have to know it exists and know it's accessible. So this is the main aim of our webinar, is to articulate the depth and breadth of the capabilities accessible to the research community in Australia. So this is a little bit of a rundown of the webinar and what we're going to be talking about. We're going to start with a little bit of housekeeping and an introduction to NCRIS and TIA and a little bit of context around the whole program. But really, I think the most important reason for this webinar is to allow the facilities and capabilities to speak and to let the community know what is available and how they can assist your research and development projects. And I'm delighted that we have three of the directors of our facilities to talk across each of our three product-focused capabilities. Then I will come back to talk about how to access these capabilities, how to find them. And then finally, there'll be an opportunity for a bit of a Q&A and to ask questions via Zoom's Q&A function throughout the session. But before we kick off, I'd like to begin by acknowledging and paying my respects to the Yagara people, the traditional custodians of the land on which many of us are gathered here today, although I recognise we are scattered far wide across the country, and pay my respects to their elders, past, present and emerging, and also to any other Aboriginal and Torres Strait Islanders present on this call. So a little bit of housekeeping. If you could ask questions all along the way via the Zoom Q&A function and not the chat window, we will address the questions at the end of the speaker's program. Secondly, if you've registered for this webinar, you'll be sent a brief survey after the webinar has finished. And I'd encourage you to take a little bit of time to fill that in and give us an idea of the audience and any feedback or areas of interest that you may have. And finally, the webinar is being recorded and will be hosted for posterity on the TIA website for a few months afterwards. So if you have to leave early or have technical difficulties or have a colleague that is interested, the content and the presentations will be available online. So let's start off with what is NCRIS. This is a bit of a helicopter view, but NCRIS stands for the National Collaborative Research Infrastructure Strategy. It's a federal government program that funds a national network of research infrastructure projects. The program supports a range of projects across, at the moment, 9 national priorities, and one of which is therapeutic development, and that's the one of which we are most active and interested in. At the end of the day, NCRIS enables access to national research infrastructure, and it's been quite well funded on a bipartisan basis for at least the last 10 years. What I do want to note that the C in NCRIS is collaborative. Now, why do I mention that? Because operating alongside us are really much an extended family of NCRIS projects that are also related to that therapeutic development priority. Now, in no particular alphabetical order, I'd like to acknowledge that we have the National Fabrication Facility that specialises in just fabrication and rapid prototyping, 3D printing, and such like. Bioplatforms Australia, let me get my laser pointer out, focuses on omics and characterization and bioanalytics. Microscopy Australia, the clue is in the name as to what they offer access to. National Imaging Facility for PET, CT and other imaging, including small animal imaging. Phenomics Australia, who specialise in development and distribution of animal models for disease, and also lately CRISPR and other genomic medicine activities. Finally, the Population Health Research Network, who operate a network of linked health data across the entire country. Now, the reason I mention these is that if we are not able to help you in your research, we are more than happy, in fact delighted, to put you in contact with any of these projects who may be able to help you. So in terms of where we position ourselves in the landscape, we have enthusiastically, evangelically adopted a scheme for articulating the development pipeline from basic biology all the way through to population health. So in general, we position ourselves or note that our facilities are most active in the area between discovery getting in towards early phase clinical or preclinical research. And you'll see this mentioned fair. We work closely with our related NCRIS projects and also with MTP Connect. We've talked to them quite a bit. They help us out with some of our assessments. So this is where we can see ourselves fitting in the scheme of things, if you like. And this is another way of putting that D1 to D4 scheme. It's a big scary diagram created by the US National Institutes of Health, I think to give politicians nightmares. But what it does do is to articulate that the development of a therapeutic from go to woe is long, it's risky, it's expensive, and very few organizations can do it alone. And in an area like Australia, it does mean that access to the technical expertise and people who've done this is invaluable, and that is what we aim to make accessible. So what does TIA do? Well, TIA Limited is a company that manages NCRIS funding on behalf of the government. But what this means in real terms is that this funding supports and enables a TIA consortium, which is what we're going to hear about a lot today, which is a group of around 24 research infrastructure facilities that are active in the area of therapeutic development in 3 broad areas: from small molecules, biologics, and cell and gene therapies. Now, we support these facilities in order that they can offer access to their particular facilities and capabilities to researchers and SMEs outside their particular host organizations. So an idea of the quanta of support here is that we have been in existence for quite a few years now, and we are currently managing around, have managed around $71 million in total of investment over the years in hard and soft infrastructure, that is people and equipment, the key facilities. Now, the reason I put this graph up is the importance of the green bars. Now, the green bars are people. Now, it's fair to say that most of our investment is directly in people. Because when you're accessing a facility supported by TIA, it's not particularly access to the equipment, it's access to the expertise and the experience and the lived experience of the facility staff and their leadership. And this is a point that you'll hear our speakers emphasize repeatedly, I think. So this is just a bit of a capture from our website where we highlight some of the management and research leadership. But overall, we fund in the region of 91 staff to various degrees, and they range across technical and scientific and management skill sets. So I'm keen to get onto our speakers from each of our capabilities, but just to give one more bit of context, we have 3 product-focused capabilities in small molecules. We encompass hit ID through to GLP tox, biologics and vaccines, and finally a group of facilities specializing in cell and gene therapies. And without further ado, I would like to hand over to our first speaker, is Emeritus Professor Marie Smith, AC, who is leading researcher in biomedical translation with international expertise in development of novel pain therapeutics. It's also the co-founder of TetraQ, another TIA facility, and inventor of patented technology that was licensed to Spinifex, and then onwards to Novartis in 2015. So she's walked the walk and talked the talk. She's also a tireless advocate for women in technology, and lastly, been a very active and long-term supporter of TIA pretty much from the beginning. So I'd very much like to invite Marie to give us a bit of a rundown of our small molecule capability.
In Australia, the majority of biomedical research is undertaken in universities and medical research institutes. And this research is rich in novel drug targets, but comparatively weak in small molecules. And so one strategy for addressing this has been formation of the small molecules set of capabilities by Therapeutic Innovation Australia. And so the small molecules platform is comprised of 5 translational research centres that have complementary expertise and know-how in small molecules discovery and development. In addition to that, there is a network of compound screening network that I won't speak about, but Stuart will speak about immediately following my presentation. So the 5 translational research facilities that I'm going to talk about are Compounds Australia, the Australian Translational Medicinal Chemistry Facility, the Centre for Drug Candidate Optimisation, the Centre for Integrated Preclinical Drug Development, and TetraQ. So Compounds Australia is a national facility that stores various open access molecule collections. All up, there are more than 76,000 compounds, comprising 33,000 compounds in the open scaffold collection, encompassing 1,200 core scaffold groups, more than 3,000 compounds in the open drug collection, comprising the microsource spectrum FDA collection, the select kinase collection, the select epigenetics collection. In addition, there are more than 40,000 compounds in the open academic collection, comprising 1,000 from the University of Adelaide fragment collection, 9,000 from the academic researchers around Australia, and more than 30,000 compounds from the CSIRO. Apart from the small molecules, there's also the Nature Bank collection, which comprises more than 18,000 extracts and 90,000 fractions from biota within Australia. If you are supplying compounds for storage and curation at Compounds Australia, you need to supply them in vials, deep wells, and microtubes. The Compounds Australia has state-of-the-art facilities for storage and curation of samples, as well as for plating samples to satisfy researcher requests in one of multiple formats for high throughput screening. And so these microwell plates will then be dispatched to the high throughput screening facility of the researcher's choice. This slide provides an overview of the entities that have compounds stored at Compounds Australia and that have accessed compounds at Compounds Australia. So you can see it truly is a national facility. Next, I'm going to talk to you about the Australian Translational Medicinal Chemistry Facility and the Centre for Drug Candidate Optimisation that work hand in glove to bring molecules forward that are more drug-like than what you get from a typical hit out of a high-throughput screening campaign. So the Australian Translational Medicinal Chemistry Facility is a national drug discovery capability that applies medicinal chemistry expertise to translate biomedical research towards clinical relevance. It fills the gap in the translational pipeline, linking early stage research in disease biology to the targeted clinical use where there is then inherent commercial value. The ATMCF scientists collaborate with researchers to identify and synthesize bioactive molecules into more drug-like molecules that we call drug candidates by making them more drug-like, potent, selective and efficacious, and importantly, in a clear space for licensing. The Australian Translational Medicinal Chemistry Facility is home to various early stage multidisciplinary drug discovery capabilities, including the planning of drug discovery programs, designing studies for structure-activity relationship definition, finding novel strategies via literature reviews and advanced chemistry skills, high-throughput screening hit selections, fragment-based drug discovery approaches, as well as mechanism and structure-based drug design strategies. So with recent support from the TIA, the ATMCF has launched APHID, the APHID capability. So what APHID is Action Plan for Hit Identification. So this is using the hit triage expertise of the ATMCF scientists to identify compounds from the hits that are tractable against their specific targets by using electronic structure-activity relationship approaches to mine the high-throughput screening deck, analysing, identifying and selecting analogues worthy of follow-up, grouping hits with particular commonalities into substructural clusters for purposes of refining the electronic substructure and pharmacophore searching, identifying structural regions of compounds where modifications may be favourable or unfavourable, assimilating all of these analyses for creation of virtual structure-activity relationship sets, assessing the feasibility of purchasing the design sets of analogues, and forecasting the cost and time efficiency of sourcing the analogues of interest. As I said, the ATMCF works hand in glove with the Centre for Drug Candidate Optimisation. And this is very important for defining the absorption, distribution, metabolism, elimination, and pharmacokinetic properties of the small molecules from drug discovery. Depending upon the stage of development, techniques or assays that will be used in silico, in vitro, and in vivo assays. I'll talk to them on the next slide. The CDCO has well-developed systems with 18 years experience, and their work is conducted in accordance with the requirements of the ISO 9001 quality management system. Over the 18 years, CDCO scientists have collaborated with numerous researchers and small and large companies, and have identified 34 novel candidates that were progressed into clinical development by partner organisations. So very well credentialed centre. And overall, the aim is to get the right concentration to the right place in the body for the right duration to foster or enable efficacy and safety testing of the test item. So the assays that are used at the CDCO fall into two main categories depending upon the stage of development. So from hit to lead optimisation, the CDCO scientists link in silico and in vitro data to the in vivo pharmacokinetic profiles, identify limiting features in the structures that impact oral bioavailability, define the clearance mechanisms and understand the biodistribution of the molecules, identify metabolites and potential safety risks, such as inhibition of cytochrome P450 metabolism or the formation of reactive metabolites. And by linking pharmacokinetics to efficacy in animal disease models, it's then possible to define pharmacokinetic, pharmacodynamic relationships. With lead optimization to drug candidate selection, this element of the work gives good mechanistic understanding of pharmacokinetics and bioavailability, identifies potential species differences in metabolite profile, which is very important in drug development, defines expected safety margins based upon the pharmacokinetic profiles, identifies appropriate phase 1 formulation approaches, and these data can be used to predict the human pharmacokinetic profile and the likely efficacious dose. So in other words, the preclinical pharmacokinetic data will inform the human pharmacokinetic data and dose predictions for early phase clinical trials. So to do this, the CDCO has a wide range of validated assays to guide drug discovery. And core activities include physicochemical profiling, chemical and metabolic stability, membrane permeability, plasma protein binding and partitioning, cytochrome P450 inhibition, pre-formulation and formulation, preclinical rodent pharmacokinetics, and bioanalysis, which supports many of those core activities. Bespoke activities include metabolite identification, pinpointing reactive metabolites, time-dependent inhibition of metabolism, reaction phenotyping, enzyme kinetic analysis, mechanistic metabolism, biliary secretion and brain distribution, solid state properties, which give important information on whether or not the drug will have a decent shelf life, preclinical pharmacokinetic, pharmacodynamic modelling, human pharmacokinetics and dose prediction, and physiologically based pharmacokinetic simulations. Next, I'm going to talk to you about the Centre for Integrated Preclinical Drug Development and TetraQ. So the Centre for Integrated Preclinical Drug Development at the University of Queensland undertakes work in rodent disease models. So we have 15 rodent pain models and 3 other disease models at this point in time. And the pain models is an area of particular expertise. For these various disease models, many of the readouts are behavioural, and so therefore the scientists conducting these studies have to be blinded to the treatment to avoid potential inadvertent researcher bias. Apart from these rodent disease models, we also have the capabilities to assess respiratory alterations such as respiratory depression, constipation, the effects of the drugs on general health and various clinical observations, collecting tissues for ex vivo investigations, and also capabilities to do in vitro efficacy assessment using various plate-based assays. We have a very large number of dosing routes that we can do, but the most popular are intraperitoneal, subcutaneous, and oral dosing by gavage. So as I mentioned, we have very highly specialised expertise in novel pain therapeutics, a portfolio of sophisticated and refined rodent pain models that mimic individual human pain conditions. All studies are conducted in a purpose-built NATA GLP recognised facility, and this makes the CYP-DD unique in Australia and rare internationally. And this is done to improve preclinical to clinical translation. And we also have the capabilities to establish other efficacy models in rodents upon request. The rodent pain models can be subclassified into 3 subclassifications. So there's 8 neuropathic pain models, 3 models of chronic inflammatory pain, and 4 models of nociceptive pain. And that's the type of pain that gets better by itself, such as pain post-surgery, resolving as the wound is heal. TetraQ is a University of Queensland research infrastructure centre. It has a fee-for-service rodent toxicology and pharmacokinetic capabilities, and the studies are done on a cost recovery basis, and single repeat dose study designs, and pharmacokinetics and toxicokinetics are derived from plasma concentration versus time data using the industry standard WIN non-WIN software package. The scope of services includes research or exploratory toxicology and pharmacokinetic studies, as well as GLP toxicology and toxicokinetic studies. The studies are managed and data collected and supported by the US FDA 21 CFR 11 compliant Preventa's software system. And pharmacokinetic analysis is supported by TetraQ bioanalytical services. So the research toxicology services are early stage toxicity studies conducted under non-GLP conditions. And what we mean by that is the work is done to the same standard as GLP studies, but not subjected to audit. And study outcomes include identification of the maximum tolerated dose of the test item. The results are used to support the design of key efficacy studies, as well as formal GLP toxicology studies. And also pharmacokinetic studies in rats and mice are used to support toxicity and efficacy study design, and capabilities include conducting studies in cannulated rats. GLP toxicology studies are also known as formal toxicology studies. They're subjected to quality assurance audit, and the data is used to support the design of first-in-human clinical trials. The test item needs to be very well characterised and manufactured typically under GMP conditions. The study design is informed by the data from both prior non-GLP exploratory toxicology studies, as well as the intended clinical use. And the study results are used to facilitate safe design of clinical studies. And very topical, a case study recently done this year was to test the UQ COVID-19 vaccine in a rodent toxicology study prior to the initiation of the phase 1 clinical trial. So who do you get in contact with? Well, overall, we have a shared business development manager, Dr. Fern Lim, and these are her contact details. And we also have the contact details of each of the facilities if you wish to contact any of the facilities directly. So thank you very much.
Thank you very much, Marie, for that. And as Marie noted, I'm just going to speak very briefly on compound library screening capacity. Okay, so brave, as Marie noted, after, if you like, after the pipeline for compound library logistics and handling, we support via a voucher scheme access to a national network of high throughput screening centers, and I just have a listing of them here, and they vary in their capability and capacity. Include specializing in antibiotics. Some of them specialize in 3D screening, and some in fragments, for example, at MIPS, and some in CRISPR screening and particular cardiac models. Now, these centers are supported by TIA via a voucher scheme, which I'll talk about a little bit later on, but I just wanted to point out that there is some support for high throughput screening available within the TIA consortium. Okay, so I'd like to move on to our next speaker and introduce Professor Trent Munro. who's the director of the National Biologics Facilities Queensland mode, and senior group leader at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. Trent has been kept very busy lately as the program director for the CEPI-funded UQ vaccine rapid response pipeline, and has been heavily involved in development of the vaccine based on molecular clamp technology discovered at University of Queensland, developed and protected by UniQuest. So without further ado, thank you very much, Trent. And so you've already way ahead of me and shared your screen. And please can tell us a little bit about the National Biologics Facility.
Great, thanks, Stuart, and thanks, Marie, too, for giving that great introduction. I think it dovetails well from here. And thanks to everyone who's joined. It's great to see a good number of people on this webinar. I guess if there's one advantage to all the craziness this year, it's we've all become very efficient at getting on shared meetings. So just want to check there's a Q&A. Okay. All right. So I'm going to talk to you about the National Biologics Facility on behalf of the nodes around the country, and I'll introduce those as we go, and also ensure that we give out the contact details for those different groups. Our role is really in enabling infrastructure for translation of biologics and vaccines. Primarily, that involves proteins and protein conjugates and engineered molecules that are produced in mammalian cells primarily. Let me just see if I can advance here. So like Marie said, I mean, really the challenge around the complexity of large molecules is simply molecular weight. And if we look at some traditional small molecules compared to large molecules, our ability to characterize and understand and produce consistently these type of proteins is challenging at a research scale, but it's even more challenging to think about how we turn these things into products. And so I think what we really want to be able to do is both help people generate material, enabling material so they can do their studies, but also help them ensure that they have something at the end of the day that can be turned potentially into a therapeutic. And I think the environment we all work in is that of one in a lab where many things are easily doable. A lot of people are able to make their protein themselves. They're able to characterize that. They're able to use it for biological assays. And the off-the-shelf solutions now are very, very good in this space. I think where things get a lot more difficult is thinking about, what is it that you need to understand in that as you transition out of a lab-based environment and hopefully one day into making kilograms or even metric tons of this material should your idea be successful. And so it's really that focus that needs to be in people's minds. And there's lots of different tools and ways of thinking about that. And I think it's an area of expertise and an area of advice that we really want to get to people early. And the reason is because you've essentially got one shot of doing it. Once you moving into clinically enabling studies, you really don't want to go back and redesign your molecule if you can help it. So this idea of getting it right from the start. So I think Stuart mentioned this at the start, and Marie as well, that we have this wealth of really high caliber research in this country across a whole range of therapeutic areas where people are developing molecules or discovering pathways that we want to interdict, which could have a really high value therapeutically. As I've said, turning that into the drug, what's the one thing that matters? And for us, it's about manufacturability, and also what kind of properties you design in. Okay. So this is going to be part of some of the new communications coming out from NBF and TIA about how these different areas that I want to talk about today, specifically for the National Biologics Facility, fit in. And so it's that same type of idea of the continuum here. If we look on the left-hand side of the screen, it's doing that research, screening through to your lead, going through proof of concept, getting your key preclinical studies done, and then moving into that clinical manufacturing. And that, from a protein production point of view, really changes depending on where you are in that continuum. obviously you're going to start with very small levels of production and trying to understand material through to things that become more scalable and are going to generate the large amounts of material you need to do. And in blue, some of the areas of focus, whether that be molecular and structural biology, protein antibody engineering, things like phage display or hybridomas, or even looking at bioconjugation for adding novel molecules onto your proteins, looking at what sort of cell production systems you're going to use and how you're going to do that, through to how you're going to do that production. You're going to use a bioreactor, you're going to use shake flasks, how are you going to characterize that material? Are you ensuring that you have the same type of tools that would be expected from a regulatory point of view to understand the attributes, the product attributes of your molecule? And then of course into the very tangible things around how you're going to generate enough material, how are you going to ensure that you can do release of that material under the required quality frameworks to enable clinical use of the material. And so the investment here in this space has now spanned more than 10 years. So there is, and I apologize, they've just started drilling here right on cue as I needed to speak in the building. There has been a significant investment in this space. And so I think highlighting to all of you exactly what's available and what you can access is really the point here. So what are we and where are we? So there's 3 major nodes of the National Biologics Facility. So there's one here in Queensland at the University of Queensland. We're based in the Australian Institute for Bioengineering and Nanotechnology here in St. Lucia. There's another facility based at UTS in Sydney, and then at CSIRO down in Melbourne, spread across 2 facilities at the moment, one in Parkville and one in Clayton. And I'll mention specifically the expansion that's going on at the Clayton facility, which is, I think, an exciting next step for the facilities for biologics. So how do the different facilities differ from each other? There's some overlap. What sort of focus areas do they have? I want to give you a general overview of that. If we start with the University of Queensland, our focus here is really on the early stage biologics discovery and development, moving on to both research and development from a next-gen bioprocess capability. At UTS, they've got a lovely facility. It's called the Biologics Innovation Facility. They're able to do 2 different things. One is the facility is set up to enable professional training. So there's a whole training center there, and it's basically set up exactly like you would see within a typical biotech company. So there's very few facilities around like this where you can go and access equipment, access training schemes, etc. We'll get into a little bit of that. And they're also able to offer a process development and small-scale manufacturing by using that same facility. At CSIRO, the focus is on biologics production, both from a research scale all the way through to cGMP, doing process development and bioconjugation and biophysics as well. And what you can see here, I'll mention, is what the new facility is going to look like that's out there at Clayton. All right, so just to go through some specific capabilities, I think, some of this may be very relevant. Some of it you may not fully appreciate what these bits of kit are, but, I think the important thing, the takeaway is that from a biologics development point of view, this is really what you would normally find in a full commercial, either contract manufacturing organization or within a commercial biotech company. So here in Queensland, as I said, antibody discovery, protein engineering, and molecular biology, really allowing you to take your lead molecule from lab-based processes into something that can be then used to develop a therapeutic. We specialize also in cell line development, which is the key to having a scalable process, and that essentially is putting your gene into a mammalian cell production system that lets you have high-level production, something that lets you make stable cell lines, master cell banks, etc., that you can transfer for the full lifetime of your development program. We focus both on the bioprocess side, but also on the analytical development side, so both the production and characterization. As part of the antibody discovery part of the work, we do have a standalone phage display facility allowing for antibody discovery. That's actually based in a separate building to keep all of the phage activities separate from the biologics work that we do. In terms of the actual facility, we have general lab space, but we also have 200 square meters of PC2 large-scale facility, which is completely segregated from all the rest of the lab space here at AIBN, airlock-based entry, separate access controls, etc. In terms of capabilities, on the upstream side, we can do from large numbers of shake flasks all the way up to 50-litre production. We have GE wave systems, GE 10-litre XDR stirred tank bioreactor, and also a 50-litre Sartorius stirred tank bioreactor. Both of the larger bioreactors are using single-use technology, so it's latest generation in terms of ensuring that we're matching what's out there in industry. On the downstream side, it's what you'd expect. So we have millipore equipment for allowing clarification with depth filtration, also various other equipment for doing other types of filtration through the process. For purification, we have actor platforms all the way up to a pilot, actor pilot, which is fully sanitizable for high quality material generation. Analytically, we've been investing in that over the last year. So we've got the full suite of analytical equipment, whether that be capillary electrophoresis techniques, whether that be UPLC. And we also have a node of the Bioplatforms Australia group here, which has a massive mass spec facility with really high-end, high sensitivity mass spectrometry capability, as well as another facility that's UQ owned. So a whole full suite of development, just to show you what that actually looks like in general terms. So this is an image of the cell line development bay. This is one new piece of equipment we have here for doing rapid single cell isolation. We also have traditional methods of doing single cell isolation like flow cytometry. Here is an image of the actual production suite. You can see the Sartorius reactor here, an example of these guys going through a depth filtration process. Down at CSIRO, primary capabilities around scale up and bioprocess development. They really span the full range here, from small amounts of material right up to tens to 100 grams, able to do process development and optimization. Again, on the upstream side, they have a number of production vessels to suit what the need of the program is. And then as part of the new facility, they'll be having a 200-litre stirred tank reactor. So that's really key for scale up, especially for clinical needs. And it's hoped that that multi-purpose GMP facility will be coming online next year. So again, there's an existing shell. This is part at the Clayton CSIRO campus, which is co-located with Monash. So this is what that somewhat artist's rendition of what that facility will look like. So that's well underway. And again, just some images of the actual facility and the type of capabilities they have. So within the existing facility before that's transitioned over to the new facility. Down at the Biologics Innovation Facility at UTS, they were awarded some internal funds, which really let them build this state-of-the-art facility they have. So 430 square meters of combined PC2 clean room, as well as Class BD suites that can span across research, training, and early stage manufacturing. Through a partnership with Cytiva, they've really also got a large suite of high-end equipment, both on the upstream side and the bioreactors they have, and the downstream side as well. So this is what the facility looks like. So there's a very nice viewing corridor. You can walk around, so that's ideal for training. You can look right into the facility. This is just an image of one of the bioreactors they have down there. And that's one of the downstream skids. All right, so I've put the contact details here of the key contacts across the 3 nodes. So myself here at University of Queensland, Edwin Huang at UTS, and also Susie Nielsen at CSIRO. So please feel free to reach out to any of us. In terms of how we support, depending on the work and how it's funded and which facility, as Stuart mentioned, that's all a little bit different. But generally, because of the support we get from NCRIS, we're able to provide, really supported research, and that's the goal. Is not so much, what it's going to cost, but how we can enable the research that you have and how we can enable you to translate that research. Your access to the full spectrum of capabilities from molecule discovery through to clinical grade material, which is rare, and also that advice and guidance on product development, which is so important. I'll finish there and hand back, but just to say that, over the years, there's some old logos on here and some newer ones. This is just some of the folks that we've been able to help, and I think many of us are proud now that a number of the programs have moved out of development phase and really into clinical phase. I think that speaks to the capability we have. We're looking forward to continuing the roadmap and continuing and expand the types of therapeutic modalities we're able to offer. I think Australia's definitely identified this in general as a gap for sovereign manufacturing. We're only 3 nodes. I think we'd like to see an expansion both in the people and capability to support the large number of programs that are approaching us and wanting help. So hopefully that was informative. Looking forward to trying to help with projects moving forward. Thanks for listening.
Thank you, Trent. Thank you very much indeed for that. Now, I think it's just a reminder to keep the questions coming in, and we'll do our best to answer them at the end. If we do run out of time, we will be more than happy to answer questions offline or take them on notice, as they say. So, but not too much ado, I'd like to welcome our final capability speaker. There's Associate Professor Marion Sturm of Cell and Tissue Therapies WA. Now, Marion's been involved with TIA for many, many years, possibly from the very beginning, and particularly in the development and translation of cell and tissue-based therapies to the clinical setting directly, particularly mesenchymal stromal cells, CAR T cells, and tissue engineering of bone. And when I recall visiting a couple of years ago, seeing the pretty cool stuff in the tissue engineering that was happening, the basement somewhere at Royal Perth Hospital. Mary's been a member of the TJ Advisory Committee on Biologics since its organisation, director of a biocompany, Isopogen, and is non-executive director of Stem Cells Limited. So I'm very keen to hear a bit more about the cell and gene facilities that come under the TIA's capabilities. So Marion, if you'd like to share the screen.
Okay. Thank you very much, Stuart. And it's great to have a lot of people on the call to hear about the scope of all of the capabilities that come under the TIA. The cell and gene therapies capabilities made up of 5 facilities spread across the 4 states. Collectively, the facilities have experience in various types of cell and gene therapies, including, as Stuart mentioned, tissue engineering from our own site, gene therapy vectors, CAR T cells, which are genetically modified T cells to fight cancer, immune and non-immune cells, and islet cells for transplantation. Since many of the facilities are embedded in hospitals, oversight from the collection of the starting material or tissue through to the clinical administration of the products can be part of the facility's activities, and also includes institutional and commercial clinical trial work associated with these emerging therapies. I might add a lot of these therapies in development and coming through now, they've been driven by unmet clinical needs, and so that interface in the hospital is so important because the clinicians come down and discuss the problems with the various facilities, and so some of the research is initiated basically through clinician demand. Manufacture of therapeutic goods for clinical application in Australia is overseen by the Therapeutic Goods Administration, the TGA, and it ensures the safety of clinical products. And so a critical function of all the facilities is a rigorous quality management system and demonstration of compliance to regulatory codes and standards. And as a result, most of the facilities are either TGA licensed or accredited by other bodies, or at least working towards accreditation. Now, as I explained, since the facilities are at the clinical interface, they all have great skill and experience in therapeutic manufacture and product delivery. They're absolutely critical to the translation of health discoveries. and I always think of them as the gateways to the clinic for the cell and gene therapy discoveries. Each of the facilities have sent a couple of slides that they'd like me to discuss. So I'm going to briefly introduce each of the cell and gene therapies. And you'll be able to see that even though each of them have got specialities in the type of product they're focused on, there are many commonalities between us, and collaboratively we can drive the development of the cell and gene therapy sector. Now, I might just minimise that. Now, the first facility is my own facility, Cell and Tissue Therapies WA, which is located within the Royal Perth Hospital in Perth, and of which I'm the director. It's been licensed by the TGA for the manufacture of biotherapeutics since its construction and commissioning in 2006, and also holds international accreditation, which is called FACT, and also NADA accredited. Now, our activity framework is based around clinical service delivery. And as part of that, we have an extensive quality management system for the facility, the manufacturing facility, as well as clinical quality management system, since we look after a couple of clinical programs. And as I said, we're driven by unmet clinical needs. So we've got a research and development arm, and NCRIS, and the money coming through the TIA has been imperative in the progress of our R&D. And we're able to assist the translation of products in development to be GMP compliant, TGA accredited, and assist in the evaluation through clinical trials. And CTTWA sponsors, supports and consults investigational driven and industry sponsored clinical trials. CTTWA also provides a bit of third party manufacturing activity within our facilities capability. And the types of products that we manufacture are firstly for supportive care of patients, and those patients that are undergoing autologous bone marrow transplant. And currently it also includes a genetically modified product for the correction of an inherited disorder, cytotoxic therapy for allogeneic bone marrow transplant and the development of allo and auto CAR-Ts, immunomodulation, and that's with mesenchymal stromal cells for a variety of immune disorders. And as was alluded to, we do a bit in the tissue regeneration area. Now, CTTWA has 5 ISO 7 clean rooms, all outfitted with standard and some specialised equipment. We have support areas that provide testing service for patients and products, and cryogenic storage areas for short and long-term storage of products. Access is also available to the adjacent research and development laboratories and all their equipment. So we have quite a nice setup where the R&D takes place outside of the clean room, and when it gets to a point that we can line it up to be GMP compliant, it can then transfer into the manufacturing facility. Now in Queensland, there is QGen Cell Therapeutics, which specialises in adoptive T-cell therapy, and it has more than 20 years experience in manufacturing, in clinical trials, and in assisting researchers in developing their GMP processes. It participates in both investigator-driven and industry-sponsored clinical trials also, and I think you'll find all of the facilities all have that activity in common. QGen is TGA licensed to manufacture for phase 1, 2 and 3 clinical trials in Australia, and is manufacturing for international early phase clinical trials. QGen's activities cover the spectrum from product development, from collection of starting material through manufacture to distribution of products for clinical trial evaluation. Their experience goes beyond adoptive T-cell manufacture and also covers the manufacture of mesenchymal stromal cells, iPS cells, cell lines, and other types of things. They also have on-site GMP accredited testing platforms for rapid release of manufactured products, and this includes sterility testing and mycoplasma testing, immunophenotyping, potency and release testing, and other essential testing criteria. QGen is part of the QIMR Berghofer and has 12 ISO 7 clean rooms and 3 ISO 8 clean rooms, as well as a PC3, BC3 containment suite. It has dedicated controlled areas for raw materials and finished products and looks a fabulous facility. Now, the Sydney Cell and Gene Therapy capabilities is located at the Westmead campus and is comprised of 2 PC2 clean room facilities, each with 3 clean rooms. And these facilities provide 3 main functions. Firstly, one facility is used for the manufacture of cell therapies for investigator-initiated clinical trials activities, and this is in cytotoxic T lymphocyte manufacture, CAR T-cell therapy in early phase trials, and allogeneic islet cell transplant program, which is in phase 3 clinical trial. Secondly, the other facility is used to dispense PC2 products such as phage therapies for bacterial infections and gene therapies, in particular for spinal muscular atrophy. And these are being used in phase 3 trial, and a number of patients have already treated with the product. Sydney Cell and Gene Therapy also have small-scale viral vector manufacturing that has recently received $25 million investment by the New South Wales Department of Health, and to expand their manufacturing capacity to a much larger volume, which is terrific for them. Now, also in New South Wales, there's the Cell and Molecular Therapies facility at the Royal Prince Alfred Hospital, which is headed up by John Rasco. It too has had extensive experience in cell and gene therapies across all stages of product development, enabling translation of research to the clinic. The clean room facility is compliant to GMP, good manufacturing practice, and ISO standards, and is PC2 certified and meets the needs of clinical academic researchers and international industry partners. Royal RPA, Cell and Molecular Therapies, has dedicated clinical trial teams integrated with the GMP facility, and that enables the collection, storage, formulation and dispensing of investigational products for phase 1, 2 and 3 industry and investigator-initiated studies. And they operate in accordance with TGA, FACT and IGTR standards right through from the collection to the product administration and delivery process. Finally, in Victoria, the Peter MacCallum Cancer Centre, which is a centre of excellence in cellular immunotherapy, has an affiliated cell and gene therapy GMP manufacturing facility. It has been a fee-for-service facility since 2003 and has been a leader in the manufacture of CAR T cells since 2006. The facility has, of course, robust quality systems and is TGA licensed for T-cell manufacture. Like many of the other facilities, the Peter Mac offers a complete manufacturing process development and deployment service and needle-to-needle control with clinical integration. Its services support projects from translation and first-in-human clinical trials all the way through to commercial programs. They have expertise regionally and globally, and access to the US, Europe, Japanese, Australian, Korean, Chinese, and other Asian markets. The GMP facility obtained a TGA licence for T-cell manufacture in 2019, and the licence covers collection, manufacture, testing, storage, and release for supply of any T-cell product, including CAR T-cell therapies. And it was the first Australian entity in Australia to obtain a licence for CAR T-cells. The TGA licence means that the clients can be assured that products are manufactured in compliance with commercial regulations in Australia, and it significantly de-risks the development process for this type of product. The licence also supports supply to multiple jurisdictions and extends to both clinical trials and commercial supply. And Peter Mac's GMP facility has 10 clean rooms that are TGA licensed, BSL2, QC lab equipped with various testing platforms. The quality management system covers collection, which is mostly apheresis, and also has cryopreservation functions. The facility is currently undergoing extensive expansion, which includes the building of 3 commercial scale clean rooms with the capacity of commercial supply of several products simultaneously, which will be functional in 2022. So it's a great piece of infrastructure for Australia. So you can see from the brief descriptions I've given of the facilities and their activities that in Australia we have a strong and developing capability across diverse clinical product manufacturing. One of the, as Stuart said, one of the major investments of the TIA in our capability isn't the bricks and mortar, though we've had a bit of equipment, but it's actually in the staffing of our facilities. A major part of the infrastructure strength, I believe, lies in the trained and highly skilled workforce, not just CTTWA staff that are pictured here, but for all the facilities. From facility operations, manufacturing, testing to quality and regulatory compliance, the workforce is highly specialised, and maintenance and training of these skilled individuals, I think, is a priority for all of the facilities. So I've given you a bit of a snapshot of what's available within our capability, and I encourage any researchers in our sector that are interested in the cell and tissue therapies to seek out and take advantage of the facilities early in their product development pipeline. As Trent said, getting it right from the start can make the whole journey so much easier. And I'm sure that all of us in the cell and gene therapy capability would welcome any researchers or any people interested in development of commercial developers or academic developers in coming to us and making use of our great expertise, because actually manufacturing these products for clinical application, there's many regulatory hurdles that have to be overcome, and you really need people that are of the high knowledge in the area. Thank you.
Okay, so thank you very much for that, Marion. I very much appreciate words on that. So now I'd very briefly like to touch on a few things, how to access and how to find TIA facilities. So I'll just take another IT risk again and share my screen, and I hope it's coming up properly. So just to note that we've heard quite a bit about the various facilities. I just want to present a little bit of metrics and be a bit of a show off really into some of the metrics and that we've amassed from the various facilities over the last year and a half really, since this 3 capability structure started for TIA. So we have quite a few projects in progress, and just looking at the graph here of where the projects currently are, you can see that they roughly align to where we position ourselves, or where we see ourselves in the post-discovery, hit lead identification, preclinical develop, up to mostly the T1 and early stage clinical trials. So this information, it's an interactive dashboard. If you're interested in exploring it a bit more, just hit our website, to the dashboard. You can click a few buttons and see exactly where various projects are. But I'd just like to very briefly talk about how to find the facilities and contact them. There's quite a few ways. Marie and specifically laid out the various contacts that can be made to make contact with the facility. So you can contact us directly, either by email or our contact page. You can give us a call up here in Brisbane, or myself. You can, of course, go directly to the facilities. We are in no way a gatekeeper, so feel free to reach out to them directly. On the right here, we also have our pipeline navigator, which we have a listing of various facilities around the country, not just ours, but across the NCRIS network. And this is also accessible via our website. So Trent mentioned there's a tiered, generally tiered access, tiered pricing model specific to each facility for research users, SME and full pharma. To get more of a handle on quotes and things, I suggest you need to reach out. We'll happily put you in touch with the facility managers to discuss budgets and costs and such like. I do want to briefly touch on the Pipeline Accelerator Scheme, which we've been running for the last year or so. If you're not aware of this, this is a voucher-based scheme that allows an additional subsidy to access to various facilities, including the high throughput screening network that I mentioned earlier on. The voucher value ranges between a $5,000 to $20,000 tier and also a $50,000 tier. Currently, we have about 44, well, we have exactly 44 projects that have been supported. Most of them are still in progress. And I note that from the participants list, we have a few recipients dialing in. So shout out to them. Thus far, we've allocated about $1.6 million. So it's not a huge amount of money, but it's focused on driving access to our facilities. It's open to SMEs, MRIs, universities, also to international groups as well. We are running schemes every 6 months. We're shortly going to announce the results of one scheme, and our next round will open early March of next year and will close at the end of April. So now is probably a good time to make contact with the facility if you're looking to apply for an accelerator scheme voucher. And you can subscribe to our newsletter for announcements on what's on the guidelines, when they'll be available, and general dates and times. So I'd now like to kind of finish the remarks and go over to any questions and discussions. And thank you for those who've put in the Q&A. We'll try and answer them now. But just thank you for your time and your attendance. Subscribe to our newsletter and we'll let you know what's happening with TIA into the future. But now I'd like to hand over to Stella Clark, who's going to help me to facilitate any questions and answers that people might have. So,
okay, thanks, Stuart. So we have got a few questions. I'm going to go around the facilities. So I'll start out with Marie. And one question, Marie, was how do or can the small molecule capabilities work with international biopharma companies that may be seeking R&D partners in Australia?
In the first instance, it would be worthwhile contacting the business development manager, Dr. Fern Lim, and she has the contact details of everybody. What happens then is if it was, say, for a toxicology study, Fern would refer the person or the company to Peter Tapley or Annette Dexter at Tetra Q, and then Tetra Q would take over. So the business development manager puts the people who are inquiring in direct contact with those who can help.
Great, thanks, Maria. And I guess it's the same for whoever you are, whether you're international or Australian.
Yes,
the best way now is to go through Fern, and her details are on the TIA website.
Yeah.
Thank you. So a question for Trent. Are all the biologics facilities producing mammalian cells? And what about the production of microbial or insect cell expression systems?
Yeah, it's a really good question, Stella. So definitely the focus for us here in Queensland is at the biologic facilities primarily around mammalian cell production systems, and that's really because trying to do multiple host cell systems would be very demanding. We just don't have the resources to do it. But in saying that, and UTS, their focus is also mostly mammalian. In saying that, our CSIRO colleagues have a wealth of experience in various microbial systems and have some capability there. I think it's an important thing to keep an eye on moving forward to make sure that we can service those needs. It's even internationally, it's getting difficult to get really high quality clinical enabling material across some of those areas. I think Biology asked the question, so there's another facility that he's part of here in Queensland called the Protein Expression Facility, which can do high quality production across a range of different host cell systems, generally not clinically enabling, but definitely for research or preclinical, that's their speciality. Stuart and the rest of the team in the biologics group have been talking about how we expand and where are the key gaps for the next roadmap and what are the researchers wanting. So I would just say if people are out there are finding difficulty accessing some of those things, please let us know. There are other things orthogonal to that, things like, if RNA vaccines take over the world, we don't really have a big capability in Australia for doing that right now. So, there are other things that we're thinking about. So it's a really good question, really important. And so we would like to hear more from researchers about their needs.
Great, thanks, Trent. We had a question that came in earlier, Stuart, and I think you told me that it's in the CGT line, but there's somebody else who's going to answer that. So it was about a TGA license for T-cell manufacturer, which Marion mentioned from the Peter Mac capability. What does this mean for Australian researchers or SMEs with an early phase T-cell product?
Yep. So this is Nathan Smith from the Peter Mac Center of Excellence. Can you hear me?
Yep.
Yes. Yeah. Okay. The T-cell license, I think, and there are some, I know that there are other licenses that are held by some of the other cell and gene therapy facilities as well. What those licenses do is really de-risk the manufacture, like what it means is that the quality systems that are in place at the different facilities have been vetted by the TGA and vetted by the regulatory agencies, have been audited and are they've given their endorsement and licensure to this to enable and to ensure that the products that are being made by those facilities are safe for use in those clinical trials and everything like that. I think each of the facilities has a different approach, has a different quality system, and has a different method of bringing in and translating products from research into clinical trials. and each of those has been, reviewed by the TGA and approved. I think the Center of Excellence, the Peter Mac Center of Excellence, stands out a little bit in the fact that our TGA license covers commercial supply, so there is a little bit of a higher burden on that, but that really doesn't impact a lot of the translational programs that you're going to see early on. What it does mean is as you move down the line towards commercialization, there are options within Australia to be able to support that the TGA has vetted and has approved of.
Great, thanks, Nathan. Just circling back to Marie, a question was asked whether there are any labs with the capability to conduct GLP tox in either dogs or monkeys in Australia?
Not that I'm aware of. And so typically, so TetraQ, for example, can put you in direct contact with international providers of monkey and dog tox. So they have the relationships there.
Oh, you can certainly find the information.
Yeah.
Great, thank you.
Yeah.
And whilst you're there, another question about CIPDD. Do they have any plans to develop safety pharmacology study capability, since it has neuro behavioural and respiratory expertise and GLP status?
Sorry, I missed the first part of the question.
I'm going to read it out properly from here. If the CIPDD has a plan to develop safety pharmacology study capability.
Okay.
Yeah.
Yeah, not at this stage. There's not high demand for it here in Australia. So in terms of CNS, that wouldn't be difficult, but we haven't done it at this point in time. Respiratory is very, very specialised, so probably no in terms of doing that.
Right, thanks, Marie. A question for Marion. Whether any of the CGT groups currently have the ability to manufacture clinical grade lentiviral vectors?
I don't think so. I mean,
anybody else on the who's participating?
Yeah, the Westmead might be the best to handle it, but I'm fairly certain that we don't at this stage.
If there is anybody in listening who wants to raise their hand who thinks they might be able to answer, oh, somebody has, fantastic. It's John Rasco. Thank you, John. Thank you for joining us. Can you shed any light on this specific question?
Oh, hi, everyone. It's been a delight to hear all of these presentations. Thrilling to know how extensive Australian expertise and infrastructure is facilitated by TIA. Lentivirus manufacture for clinical grade production is not available currently in Australia to the very best of my knowledge, and no one has yet done it. No one's funded to do clinical grade production of lentiviral vector production, but there are many places, in fact, probably dozens of discovery research labs that are manufacturing lentivirus. We've been manufacturing lentivirus for as long as it's been available, towards 2 decades. And certainly retroviruses equally, which I would emphasize are also being used in clinical delivery of gene therapies for CAR-Ts, as well as hematopoietic stem cells, but much more importantly, CAR-Ts, because lentiviruses are safer in the context of hematopoietic stem cells. That said, the facility at Westmead has got AAV manufacture funded, and they do have a very small facility, but this is, I think, in product development rather than in clinical grade vector production. And we'd be in a similar ballpark using either G-Rex or the Wave or Zuri reactors for small grade production, but it'd probably be in process development and preclinical use that most people would be interested, emphasizing that can be the case in phase 1 clinical studies. You don't need to use GMP grade facilities, although it needs to be as close to a GMP as possible, and the TGA would recognize that. Thanks.
Great, thanks, John. I think that's all the questions we have, Stuart.
Yes, but I think there's some additional people who'd like to, no, some hands have gone down. hang on, no, sorry. I think Margot Latham of Sydney Cell and Gene Therapy, I think would like to add to that. So Margot.
Thanks very much, Stuart. Just to add to what John was saying, we are in the process of developing both Lenti and AAV clinical grade vector capabilities. We're hopeful that, as John said, there's no requirement for GMP for phase 1 trials with Lenti in Australia. There is scope for manufacturing of Lenti in that capacity, but in terms of GMP grade Lenti, we're probably, I'd say, 12 months away from having clinical grade Lenti available from that facility.
Thank you, Margaret. Thank you for that follow-up. Okay, Stella, back to you.
Okay, yep, I think that's all the questions answered, unless somebody wants to quickly type something in. I've just seen something saying Cytiva also offer clinical Lenti manufacture offshore. David Marbon, thank you. And I know Jonathan Bale had a cheeky question about international pharma getting R&D tax credits if they have an Australian node. Not sure about that.
I'm not an expert on this, but my understanding is that as long as a pharma company has a registered address and operation in Australia, then they would be able to access that benefit to some degree on research undertaken within Australia. But I'd be happy to follow up offline on that. I think Margot just had one more thing to add. Margot, you had your hand up? Maybe.
I think.
Maybe not. Okay, well, if there's nothing else from anybody, we're getting towards our 90-minute time slot. I'd like people to be able to get on with the rest of their days. And just like to take this opportunity to thank everybody involved in the presentations on this webinar, Trent and Marie and Marion and Stella for shepherding some questions, and also everybody who attended and for your questions as well. Just reminding you that you should receive an email from Zoom. It might be in your trash folder, but it does contain a link to a survey which only had a couple of questions, and would be very appreciative if you could follow it up, if you could complete it rather. It gives us an idea of areas of interest for future events like this, and also the demographics, if you like, of the audience today. So with that, I'd like to say thank you very much, everybody, and hope you have a great rest of the day. And Merry Christmas.